So welcome to the second core lecture on dynamical field theory on selection decisions. I begin by reminding you of what we did last week. So we talked about the different attractor states in neurodynamical fields, essentially the self-stabilized peaks that are maintained by interaction versus the subthreshold activation patterns that are really just low-pass filters of input. In the dynamic field theory, this is really a qualitative difference. Most of the kind of solutions you're familiar with from forward neural networks would be of that second category, right? It's just input-driven solutions in a sense. And then we talked about the detection and reverse detection instability, and I elaborated a lot on that. And I already showed you in the simulations the selection capacity of the fields, and I'll elaborate on that today. I showed you working memory and the boost-driven, there's a type of boost-driven detection instability. And so what we elaborated on last week was the detection instability, which will accompany us, especially in this higher cognition part. So it turns out when we're trying to do real cognition, sequences of thoughts and actions, that's a critical thing that really makes switches from state to state. So one of those peaks forming and suppressing some other peak and forming the next and so on. It's always going through an instability. And that actually, if you understand dynamic systems theory, is critical. That the instability is important in separating the time scales. That has to do with what is called the center manifold theorem. So I'm assuming most of you don't know that, but if you happen to know these kind of things, it's a core theoretical concept behind that, why that's so important. I illustrated the detection instability in the simple psychophysics example in which people have to make a decision about whether they see something specific. This example that I recall here is the detection of motion, seeing a little dot of light moving, and the detection stability implies a bistability between the detection state, where you have positive activation, versus the non-detection state, where you only have the subthreshold solution. These two are bistable. So one behavioral signature of that bistability is hysteresis. That is, if you change parameter settings to go from a regime where it's not likely to have the percept, like low relative contrast here for these movement stimuli, into a regime where you have it's likely to have the percept. Like going up here, then you will switch into the detection state later at higher parameter values than if you come from the regime where you already have that percept and you go back into the regime where you don't. There are subtleties about how to really provide empirical proof, because there could be things like perceptual uncertainty, where you're just guessing, and maybe then you're very biased to just say whatever you previously said, and various and attentional issues, if you actually even know where to look and what to look for, and what is the perceptual alternative, and do you know when to give the response, and are you under time pressure or not? So all of these things can be handled, and there's no question that perceptual bistability does exist. So today I'm going to go for the selection property of the fields and the selection instability. So this is a diagram that summarizes again the sort of thing that I showed in the live simulation. In the fields, in single dimensional field, selection shows up through through bistability or multistability. That is the fact that you might have multiple local maxima in the input, and these local maxima would be places in which a peak could arise. So for instance, if you just presented one of those local maxima to the field, it would generate a peak there and you would go through detection. But if you have multiple local maxima, you could have a peak that is either centered on one of those maxima or on the other of those maxima. So the system is bistable, has two possible responses. And which response you will get would depend on your initial condition when that bistable regime arises. So that could mean maybe just previous switch selection decision you made, or it could mean, for instance, that these two local maxima arise not at the same time, but one of them arise a little earlier, for instance, because it's stronger, reaches threshold earlier, induces a peak, and then the second one that arises a little later doesn't have a chance to bring the system above threshold, for instance, because of that suppression here. The inhibitory interaction makes the suppression. There are limits to this bistability. That is, this system like that can become monostable if one of those inputs is totally dominant. much larger in amplitude, which in neural networks would mean that the synaptic connectivity matches the current input very well, then all the other local maxima might not be viable solutions, but can never win the competition. And therefore only one is system monostable. And if then input changes back again to a more even strength of the different local maxima, you would stay with that decision. So it has the same property of stabilizing a selection decision. So once you have initially selected something, you will tend to stay there. And there are actually some very subtle effects about that in, for instance, classification decisions, where you would stick to such decisions. So we've linked this phenomenon to change blindness under some circumstances. I decided not to go there in this lecture, but it's also an interesting topic. Okay, so today I wanted to talk a little bit about the functional significance of that, and then actually link this to a lot of work happening in sort of cognitive psychology, classical cognitive psychology. Selection decisions could be viewed as sort of the core decisions. Most people don't think about detection as a decision. It's maybe this go, no go, or, but usually people think, when they talk about decision, then they think choosing one of multiple possible outcomes. Now, formally, you could say detection versus non-detection is also some kind of decision like that. But that's actually subtle, because that means that you know that you're supposed to say now that that you detect or not detect, because not detection would be just nothing, that you don't do anything, right? And that would be the more ecological thing. In an experiment, you might tell someone, so now tell me if you saw or not, and then it looks like a selection decision, but it's like just it's actually an interesting question how to model that kind of paradigm, not go there. So selection is really between different representations within the same field. There are really different possible outcomes, and like estimating different values of a parameter, or different classification decisions that you could make. And we'll get some examples soon. So here's an illustration from Claudia Willemsik's work many years ago, in which that just illustrates that the way dynamical fields make selection decisions stabilizes these decisions. So here, the field is in some initial state. There's actually some initial pre-activation, but it's activation is below 0. And then input is provided. I think in the simulation, the input was actually symmetrical at the two locations. But by some fluctuation, this location here grows activation that is supra-threshold, a peak, and then stays in that peak, and the input continues in this simulation to be present, while the other location loses and therefore is suppressed here. And there's a lot of noise on that field, and you see that that noise doesn't come anywhere close to calling into question that decision. I demonstrated in simulation that there's an incredible level of noise you would need to bring a suppressed peak like that to threshold. It would only happen really right at the instability. It's actually interesting. The selection happens when you're going through detection at the same time. That is when you're first detecting an object, then you're also making a selection. That's a very typical situation. And the last exercise, last task of last exercise sheet actually looks exactly at that situation. One of you actually found out that one needed the selection parameter setting for that. There's a mistake in the exercise, which I explained in the discussion forum. So here on the left, this is just an illustration why that's not trivial. So for instance, another possible way of making decisions is to just read out the location at which there is maximal activation. That will be an argmax kind of operation that is quite commonly used in neural networks and other kinds of information processing machineries. And this is the plane that was put in here is just used to make it visually easier to guess what would be happening. And what you see is that if you have no interaction, this is just the field as an input low pass filter. So it just has the linear parts and no interaction. And then there would be fluctuations. In this case, the two locations have equal inputs. So just by chance, sometimes activation is a bit higher, a bit lower, and the ceiling that was put in there sort of helps to visualize that. And if you were to do threshold piercing or this argmax procedure, then you can see that at different moments in time, different locations in the field would have the maximum level of activation or would have above threshold activation. And therefore the decision would be switching back and forth, be fluctuating. This can be real. We actually did a robotic demonstration of this where the robot was supposed to select a target and we turned off the interaction, and then the robot would actually meander between the two locations as noise. This was an auditory system, which I think I might have shown you, I don't remember, or I'll show you in the embodiment section. And because of the nature of the noise with that it was the sensory input that was noisy, led very much to this kind of behavior. So that's a known problem in a lot of technical approaches to information processing. People have ad hoc algorithms that sort of implement hysteresis. For instance, have an algorithm that probes how long ago you switched your selection, and then imposes like a silent period where you can't switch again and things like that, or just some kind of flow. Essentially, algorithmic implementations of hysteresis or bistability. So the stabilization of detection decisions is again important if you think of decision making taking place under continuous exposure to information. Let's say, as contrasted to the classical picture, there will be an item of information and you make the decision, and that would be just a one-time evaluation of that information. That is an abstraction or idealization. If you think, let's say, a decision, whom to marry, and then you would get one shot at looking at some people and then decide that. Obviously, that's not a good approximation, right? You have a lot of experience over time, and you have to stabilize that decision. Now, that's a very macro kind of decision. The extreme, it's perhaps intuitive that this level of stabilization of decisions is very relevant for sensory motor behaviors, and that will be a theme in the course to see that arises also, that sort of property also arises higher at higher levels. Now, how do we study this in the lab? So what would be signatures of that? It's actually difficult. It's typical, people don't really do free decisions. So free decisions, free in the sense that you're not instructed what to do, you figure it out yourself, is part of this field of behavioral economics, where it's a question of utility trade-off and what kind of choices people make. But that typically doesn't look into this really the short-term dynamics. It's more about accumulating information and so on. Also an interesting topic. But in the kind of studies that really look at the perceptual basis for decision-making, perceptual decisions, for instance, or motor decisions, or simple cognitive decisions like classifying a word or recognizing something as a grammatical phrase or not a grammatical phrase and so on. Those are all cases where free choice is not studied very frequently. So typically there is a clear correct answer and every other answer is a mistake, an error. There's usually a component of stimulus that tells you what the correct answer is. It's called the imperative signal. And the tasks just vary how hard it is to actually figure out which the correct answer is. And that from that, you're sort of inferring something about the processes that lead to that correct answer. The main reason why that is done is empirical. It's quite difficult to get participants to behave reasonably, reproducibly, not making implicit assumptions or come up with some strategies when they're not told exactly what to do. So free choice is difficult in such tasks. For instance, if you ask people to make a saccadic eye movement and you don't tell them where to look, then they might have strategies always to look at the left upper corner or always look at certain kinds of stimuli in there based on some biases they have. It's very difficult to get reproducible behavior. And these economic kind of decisions are, again, ones where there are constraints on the decisions, like you're maximizing some reward, or you have a constraint of not spending too much money. And then you get some reproducible patterns of behavior that include a lot of variance. But in sensory motor domains, it's very difficult to get anything reasonable. So I'll give you some examples of where it is possible to do free choice. And those are examples where It's actually pretty much just two examples. One is eye movements, which are so fast and sort of involuntary that it's hard to intervene with funny strategies. And the other is babies. Babies don't get a task, they just do it spontaneously. And if you can generate a situation where they have sort of reproducible behavior, then you haven't given them an instruction, right? Because they don't actually even listen to that. Yeah. So here is a little bit an illustration of eye movements. I don't know how familiar you are with eye movements. Eye movements are very constrained. You can really only do let's say in practical terms, the kind of things you do, you can really only do saccadic eye movements or tracking. So smooth eye movements where you move your eye continuously are actually only possible in tracking. And tracking means there is an object in the world that moves, and then you can follow that object. You cannot do tracking without such a target to track. So you can't say, oh, I will move my eye now smoothly here from right to left. That's impossible. What you instead do, you make abrupt eye movements that have a very uniform kinematic shape, that are quite fast, and jump across. You can't see that if you look into the mirror, because during one of those abrupt eye movements, which we call saccade, comes from a French word, visual input is suppressed in some way, or you're not perceiving while you're moving. That would create some kind of blurry image, right? So you would only see this subsequent fixations. But if you look at someone else, and for instance, have someone read a text maybe in a large type, so you have to jump a lot, then you can actually see that person's eyes make these jumpy movements. And that's we do that all the time. We do about 3 saccades a second. Now, it's quite a lot. So on average, the fixation on a particular part of the scene is only about 300 milliseconds. Of course, varies a lot, but that's over the long run. So a lot of eye movement. So just imagine what's on your retina, right? There's a wild shifting mess on your retina. It's kind of amazing. The visual impression you have of the world is constructed from that, right? It's not on your retina that you have this visual array. So this is just an illustration from a paper that tracks. You can measure eye movements. We do that in our lab. Actually, we have an eye tracker where you can measure where people look, and then they look around. There are multiple factors that influence where people look. People tend to look at locations with a lot of visual structure, like there's this high frequency stuff here that people tend to look at. This picture is seductive that way because there's a relatively low contrast background. But there are many other factors that play a role. So it's not, looking patterns are not reproducible per se. So for instance, people are very specialized in looking at humans, look intensely here as you see at eyes and mouths and faces and so on. We're very social animals. We're always looking for cues to people's aggressive or non-aggressive state, or anything that informs us about our relationship to people. And then we tend to look at objects that have meaning to us. It also depends on the task. Can somebody ask for the bottle and you'll be looking for bottle-like objects? And so it's a long history of people trying to interpret these paths, but they are subject to so many different factors, very difficult to find any specific pattern. Eye movements during reading are very much studied. There's some regularity to those. And that reflects, that's why it is possible. It reflects the difficulty of studying free choice, right? Because it's subject to a lot of influences, but it also reflects that you are making eye movements all the time unawares. Most of you might not be aware of this at all. And therefore it is something that one can study in the lab. So in the lab you tend to study much simpler scenes. Most of the classical work was on having just 1 or 2 objects in the visual array. Some of this work is done with monkeys, and there's a lot of neurophysiology. The neurophysiology of eye movements of saccadic eye movements is the best motor neurophysiology that we have, because the machinery for this is very special. It's kind of quite possible to study that in detail. The neurophysiology of human arm movements, for instance, that are also oriented at objects in the world is much, much less well understood. So here's a caricature. This is actually redrawn from this paper, caricature of these kind of experiments. You would have, as a human or a monkey, a task of looking at a fixation cross that would be present in the visual array all the time. And then at some moment in time, for instance, in this case, 2 spots of light might be presented, or under different trials, you might have a single spot of light being presented. When you have a single spot of light being presented, you tend to spontaneously make a saccadic eye movement to that spot of light or spot of contrast. Sorry. It is, under some circumstances, almost impossible to suppress that. For instance, if this fixation point disappears when that object appears, it's pretty much impossible to suppress that saccadic eye movement. If you have a fixation cross, you can try to ignore the target. So that's the background that's always done in these studies. Now here, I'm showing already a decision situation. Let's take the bottom one first. So you have this target on the top and this target at the bottom showing up, and then you would observe that sometimes the eye shifts to this target, sometimes it shifts to that target. This is a single trial. The little crosses are individual trials. So a single trial that sort of hit the center of that. And so that could be modeled quite directly by saying that some representation of the saccadic eye endpoint that is then neurally read out by the saccadic eye movement system has to be bistable, has to sometimes make a peak here on the left and sometimes make a peak on the right. And this was our first paper on using these kind of techniques in '95, where we actually modeled this, and we modeled some behavioral signatures that were then non-trivial predictions from this kind of theory. That is how we established it. It must be something like that. I should say that in the eye movement case, there's actually really good neural evidence, and there's a group in Canada who used our model to account for data, for neural data, colliculus superior. In English, they say superior colliculus, where you can actually measure something like that. You can measure essentially directly that field. I chose to not go too deep into that. For instance, in this field, there is an interesting transition, that's sort of a subtle thing, that if the selection, the 2 fixation targets are close to each other, then you actually make a saccade that is called an averaging saccade, to make it into the middle between these 2 objects. You can imagine it's essentially when you're able to foveate both of these targets at the same time. You know that we have a fovea where we have much higher resolution vision, and you can imagine that if you can foveate both of these, there's no reason to select one of these, and that's turns out to be true. There is for instance, the sort of things that you can do is you can make one of those targets much more frequent than the other target. And then on cases when you show both, so many trials with just one target, let's say the green one, and a few trials with just the red target, and then a few trials with both targets. And then you find, for instance, that this cloud of points is attracted toward the more frequent target in this averaging situation, but not in the selection situation. That's something that the model can explain. And that's the style in which we think about that. So here I want to, let's summarize this. Here I want to show you one particular wrinkle on that, because it's an interesting insight into how theory relates to behavioral data. So a wrinkle on that is that you can actually look at the time course of saccadic selection decisions. One direct way to do that is to look at saccades that have different latencies. Latency is here the time from the presentation of the visual stimulus until you initiate the saccade. The saccade has a sort of uniform length, so it's sort of the reaction time for saccades. And there are multiple ways of how you can manipulate that. So some of this variation is just spontaneous. So if you have many trials, some faster, some slower. And so here the symbols that come from the same, have the same type, they come just across different trials. It's randomness across different trials. You can also manipulate that. For instance, you can give participants the instruction to do the saccade as fast as possible. For instance, you give them a reward when they make it fast. Or you can emphasize instructions to be accurate. And accuracy means that you have a feature that specifies one of those locations as the target and the other as the non-target. And what you see generally is that the fast early saccades tend to have a a lot of errors and sometimes have averaging. So there is saccades to intermediate targets. Here, this is the non-target, that is the target location in angular distance in visual space. So averaging or even bimodal responses, but the later saccades take like 350, 400, 500 milliseconds to initiate. They tend to be on target. They are no longer affected by the non-target. So that's an interesting time course of that. Here on top, you see if there's no alternative, that's the variance you get spontaneously, and it doesn't evolve much with time. So there's a transition from averaging to selection. How can we understand that? And that can actually be understood by the theory, but we have to extend the theory. That's why I tell you about them. This is an example of models being best when they fail, because then you get new insights. So historically, it's kind of known, but it wasn't a big time failure. But the first model I showed you would not account for that. And to account for that, we have to extend the model. And the bug in the model, why it doesn't explain that, is that in the model, the two forms of interaction, the excitatory and inhibitory, live on the same time scale. That is, the excitatory is ultimately responsible for the averaging. Averaging can be driven by input, because the input is monomodal, but it can also be emphasized by excitatory interaction, that is, so that would fill in between the two minima, would fill in the activation. So in this picture here, for instance, this is sort of just subtle in the model that there is a little bit bimodal input, but there is a peak centered over the trough. And it is centered there because the excitatory interaction sort of fills in that trough. Those in the middle get excited from left and right and neutrally in that makes that peak. So the excitatory interaction would be responsible for averaging, while the inhibitory interaction would clearly be important for selection. And so both of these happen in this model at the same time. They come from the same kernel, and there's no reason why one would occur earlier than the other. Now, that is actually not physiologically correct. And I mentioned in my neurophysics tutorial, Dale's law or Dale's principle, which says that one type of neuron only has one type of synapse on its downstream axon, makes one type of connection downstream. We're based on what kind of neurotransmitter it houses. And therefore, a neuron cannot be at the same time excitatory and inhibitory. So if we think of locations in the fields being represented by individual neurons, we know that might be more abstract, but let's just take the simple picture for now. Then it's not possible to have neurons excite their neighbors and inhibit those who are further down. And the answer how this actually can be done is well known in physiology, is so-called inhibitory interneurons. And I briefly hinted at that in this layered model of cortex in the neurophysics tutorial. That is, what happens in the brain is that if you have inhibition, then you have first from the excitatory system. The excitatory system is the one that ultimately drives behavior, because to make the muscle move, you will ultimately have to excite something, right? And so those excitatory representations that really have meaning will be exciting an inhibitory layer or a population of neurons that themselves have inhibitory synapses on their downstream targets, but that are excited by the excitatory layer. So the inhibition is being, we call this then inhibition, but it's really also neural excitation, but that has an inhibitory effect. And so the red arrow here points at the fact that when the inhibitory neurons are excited, their effect on the excitatory layer will be inhibitory. So it's a two-layer system that separates excitation and inhibition. And a system like that does actually imply something about the temporal order of processing. And I want to make this concrete, not that these equations are, that you have to do something with them, just to maybe make it not mysterious. So the excitatory layer is sort of like what we have looked at in the Amari case. We have a minus U resting level input from the outside, stimulus input, and then there's a kernel, but this is a purely excitatory kernel, because excited neurons can only excite, right? So this is only so it's the Gaussian kernel, it excites its neighbors. But the negative part here, the inhibitory part, may also have a kernel, maybe a different, typically a little bit broader kernel, but it comes from a different field, V. It doesn't come from the same field, U. And that V field, that will be a population of neurons that is inhibitory in nature, in that sense that where it shows up as input, it shows up with a negative sign. So the minus K, K being positive, reflects inhibitory synapses. The V field, other than that, has its own, the neurodynamics. Often we assume there is no direct sensory input to inhibitory layers. It's not entirely true. That does happen sometimes in the brain. And it is driven by the excitatory fields. So this is the fact that the excitatory field shows up here as an input to the inhibitory field. That is this arrow here, the green arrow that their excitatory neurons project onto the inhibitory layer. Turns out that the timescale tau of these inhibitory neurons in the brain tends to be faster than the timescale of the excitatory neurons. So in that sense, inhibition is faster than excitation. And that's different from what most neurophysiologists will say. They will say inhibition comes in late, it's late. And that's because of the ordering of this coupling, not because of the intrinsic timescale in the dynamic system sense. And it's important because the Amari equation, where we essentially arises when we eliminate this layer, when we say the inhibitory neurons are so fast that I can actually neglect their time course and just say they went to the attractor of that equation. So any given current state of U would create an attractor here. You see, attractor would mean V dot is equal to 0. The minus V makes that it's always an attractor. It's always stable. So the fixed point is V dot equals 0. Put V to the other side. So the attractor is equals to V equal H plus, the input it gets from the inhibitory field. And if you replace this here by that term, so you would put in H would be just some contribution to this. And then other than that, you put in, that H actually makes a global inhibitory component. And then you put in this term, then you see there's some kind of convolved kernel that again is convolved with the U pattern itself. Now, that's how you get the Amari equation from this so-called two-layer Amari field. There's another version of that Wilson count that I'm not going into. So what we looked at so far is sort of a limit case where we eliminate the inhibitory layer as an independent dynamical layer. That it's a good approximation when inhibition is supposed to be so fast that that nothing, it's just some time delay that is missed here. But there are cases, other cases, where that second step in the dynamics matters. First one is going to simulate that in the simulator that I started here. So this is not on the web page. This requires you to actually run the Cosivina software, which sort of looks similar to the thing you are familiar with. So here you have two fields. This is the U layer, the excitatory. This is the V layer, the inhibitory. Both have some noise. The green here on top is, again, resting level plus input. The inputs only go into the excitatory layer now. And if I did this correctly, remember, I can put in some inputs here. And then we can essentially look at detection. This is actually not doing anything on the layer. Maybe we'll see what kind of connection did I put in here. VU is 0, UU. To get from excitation to inhibition, yeah, something on. So sorry, I thought that when I load this, I thought I had that. Maybe this one. Oh, I see. I have to select. Yeah. Now, sorry, I failed to press select. So now I have should have the interaction. Oops, no. Select. No. It's here. Select. Oh, why does it not? Oh, I want to select the selection one. Yeah. It should actually set this one. Yeah, not sure why it doesn't do that. Not sure about that. No, let's try it this way. So if I put some input in here, I should get a detection instability there. There you go. So you saw a detection instability again in the sense that here this system, let's reset that the system reaches threshold, yes, and then suprathreshold activation arises. It's just a little bit here. I can make this a little bit weaker. Maybe just like that. This should probably do detection. Yeah. Now you saw very clearly that this here comes up first, and only then this comes up. And the reason for that is that only the sigmoid, the red curve, makes input to this bottom layer. And so only once a peak has been generated here can you actually generate a peak at the bottom. So let's look at that again. See, on top comes first, and then it comes on the bottom. Right? And that makes this temporal sequencing that anything the inhibition is responsible for, and is responsible for selection, comes in a little later than whatever the excitation is responsible for. There are other very interesting effects in here. I think I'm going to come back if my computer can manage to run this in the background. I think it can. Here's again this principle, that excitatory excites inhibitory, and then the inhibitory inhibits, and that creates this temporal ordering of how the effects of these different interactions arise. If you look more closely, there are actually three regimes. When you have an input very early, you just have input, right? And then when you look at first, you see it's just the input. The blue rises because of the input. Then excitatory interaction enters, and then inhibitory interaction enters. These are these three phases. And there is this is from a simulation that illustrates the thing I just showed you with the naked eye. So there is a stimulus here, bimodal stimulus. The field is initially at -10 or something, resting level, and then at the first two time steps, you see that the input is just a low-pass filter of the. The field is just low-pass filter of the input, so it's input driven, and then 25 minutes later, it's sort of been driven up here. See, it's still bimodal, and it takes some time here for the excitatory interaction to really engage. It's already engaged here at 25 because you see there's an inhibitory effect on the side. I'm not sure about that actually, if there is. Maybe. But the excitation has filled this in. And then if you take, inhibition is actually already present because it's expressing everything else. But the inhibitory effect on this local decision takes time to overcome the excitatory interaction. And you see this on this different time scale. This is a longer time scale, and we use different kind of plot, a grade level plot, where you can see that what we just looked at here was here early is bimodal, the input driven. Then here where a single peak arises, that's where the excitatory interaction dominates, and then you see that the field is one of those peaks is selected. You see that the peak actually moving to the right in this case, and in other simulations it will be moving to the left, and so that occurs on a larger time scale of here, hundreds of milliseconds, while this was dozens of milliseconds. That's sort of the temporal order. And this shows up then in an account for these data. This is from the model, but as I showed you earlier, examples of data like that, and there's actually one example in the literature that looks exactly like this. So the example I showed you previously from experiment was when there is a target here at -10 and a non-target, a distractor item. So some feature information tells participants which target to go to. And there you see that the late saccades are going to the correct target, and the early saccades are doing averaging. And if you have bistable, so you have a two-target paradigm, and you don't specify which of the targets is the right one, then you get bistability for long latencies. That is, it selects randomly one of the two, and averaging for short latencies. Now, that's only under, of course, the right conditions when both stimuli are similarly potent with similar input strength. Now, this in the model, you don't get this variation of latencies without some special tricks. This actually makes use of varying the fixation condition to make sometimes fast saccades and sometimes slow saccades. And I think I have a slide about that a little later. So that's an account for a behavioral signature in the data that shows the time course of decision making on a fast time scale, right? This is so very fast sensory motor behavior. Interesting, even at that time scale, you can sort of directly see the neural mechanisms in a way to do that. Of course, it's not neural in the sense that we would have really identified particular population of neurons that exactly do that, even though this team involving Thomas Trappenberg and experimental group, this name just escapes me, fortunately, in Canada, they have actually done much more detailed work on mechanistic foundations of that. And this is more complicated than what I'm saying here. Skip that. I wanted to use that opportunity to also briefly point out that once you have two-layer systems like this, you get also other kinds of solutions that are not part of sort of our current thinking, not fixed ones. They are oscillations. How do I get oscillations? I can remember. I think it should be very generic. Maybe fine. Is it just like this? I think the time scale is more similar. Yeah, those parameters. There should be a button for the more advanced parameters. I don't remember. It's just yeah, here we go. This does an oscillation. Yeah, you can sort of intuit why there would be an oscillation. It's this time delay, right? The excitation comes up, creates inhibition. Inhibition inhibits excitation, and so excitation goes down. And as the excitation goes down, inhibition goes down. And if inhibition goes down, excitation can come up again. And that's inhibition. So this is sometimes called that the restaurant cycle. Now, good restaurant, cheap, people go to, then it becomes expensive, people still go to, then it becomes bad quality and expensive, young people stop going to, and then it goes bankrupt, or it becomes cheap again and then improves quality. This is another story. And that's important. I mean, these have been studied in the literature actually as models of epilepsy, so dysfunctional states when whole populations oscillate like that without any differentiation, and that is a non-functional state, and there is epilepsy is a phenomenon like that. There are also oscillations of that nature called thalamocortical oscillations, where whole areas are sort of locked in a way. But more differentiated forms of this are potentially the basis for active generation of time courses. So in movement, for instance, generating active time courses, initiating a movement and then terminating it without external input to control that. And even in thinking, as we'll be seeing, you sometimes might have to trigger something that takes a certain amount of time, and then you only a certain amount of time later do you want to intervene. You give the system enough time to do something. That I need a timer for that. Like, for instance, if you make a decision, yes or no, you might have evidence for yes, but no evidence, absence of evidence for no. And so what you want is you want a system that waits long enough for a yes response to come up, and if none comes up, it says no. If it said no right away, then that wouldn't work. And so you need a timer to say how long will you wait for yes response to come before you say a no response. That's one standard model of how you do negative responses. And that requires some kind of autonomous time generation. And essentially this mechanism that is paired excitatory inhibitory populations is the mechanism that does that. There are proposals that this would happen in a very complex form. This is called liquid state machines, or it's another word for that, echo state machine networks, is the same sort of idea, which is essentially just very random connectivity in which some are excitatory, some are inhibitory, and together they shape oscillators like that then can be much more complex, and you can select a subset to make any time course you wish. But it might even occur in parametric controlled way like that in motor cortex. For instance, there's a current conceptual proposal that motor cortex is a collection of oscillators like that. And we're concretely engaged in modeling movement generation by having batteries of such oscillators, the frequency of which we can tune by these interaction parameters. as a model. In this lecture course, I'm not really going there. There's a little bit, there's a box in the book about that. Sort of an advanced topic, but that's a possible and maybe necessary extension of the whole fixed point logic that I have been emphasizing so far. This is why I put this here in parentheses, because it's not something that we're going to elaborate much in this course. Okay, check the time. Okay. So I pretty much the rest of this lecture, I want to talk a little bit about how we would be actually looking at selection decisions in the laboratory, not in free choice, but in situations where there is an imperative signal. And I wanted to talk about two paradigms. First, the classical reaction time paradigm, sort of the dominant paradigm of essentially most of cognitive psychology to this day. Is a very much used kind of paradigm. Information processing story for that I'll briefly mention, and then see how we can make sense of it. And then I'll show you what we call the time movement initiation paradigm, which is sort of a twist on that, that more directly observes really time courses of underlying activations. So let's first look at the reaction time paradigm. So in the reaction time paradigm, you get a stimulus and you make a response, and the time difference between the stimulus and when you initiate your response, it's called the reaction time, and that is what's being measured. The response may take different amounts of time. You make a movement and so on, and that is usually discarded. You really want to see when it starts, and then the actual movement might be just a question of the motor system and so on. Right? So sometimes people talk about response time, and that can include the response. Sometimes it doesn't. It's a bit fuzzy in the literature if response time is the same as reaction time or not. So technically it's not, but sometimes people use it as a synonym for response to reaction time. So you're not interested in the details of the unfolding behavior. It's just when are you ready to make it? And the idea would be that that is informative about you having acquired enough information to make the response. In a little while, I'll show you that that is actually not 100% true, because you can already start responding before you're ready, and you can update while you're doing that. So, but everything else being equal, if you affect if this is relatively uniform and you just manipulate somehow this interval, then you measure something about the process of preparing that decision. Now, the idea of these classical experiments is that you vary somehow the difficulty of computing the response from the goal signal. How hard is that computation? It's a complex computation, would take a lot of time, less complex, less time. And if you phrase everything in terms of information processing, then it is, the amount of information that needs to be processed. You can see how that would make sense. Now, allude to experiments where you really vary the bit amount of information that needs to be processed in some abstract description. And then if that affects reaction time, then you consider that evidence for this information bottleneck. That it's a question of your capacity to how much information you can process per unit time. So that's where this came from as a theoretical idea. Even though the paradigm is very old, goes back to Donder's, even older, I don't remember who the first was, but the various psychologists already used something like that with a less formalized kind of intuition. Now, this all is based on knowing what the possible responses are, something that is sometimes not emphasized enough. So in this information theoretical analysis, you have to say, how much information? Well, that depends on the task. If it's always just two choices, then you just have to figure out which of the two. Even the objects might be, more or less complex, but if it's just two objects, you just have a binary class classification task, and it's in that sense, at some behavioral levels, it's a one-bit decision. So psychologists call this task set, and it's not always sufficiently discussed. So here I'm suggesting that it will be a two-choice task where you have two very clearly different objects, and you reach for one of those, and there's a cue that really shows you the size of or the shape of the object, and then it will be very easy to do that. And of course, typically people do tasks that are a little more subtle than that. And I invite you, in fact, in the exercise to think through one paradigm like that, the Simon effect, as a subtlety of how this task set matters. So the task set is very important. So it's actually the reaction time studies are all relative to a task set. This becomes very clear when you do this with animals, for instance, or monkey research. There you have to train the animals to understand the task set, understand what the task is, right? You can't tell the monkey what to do, so you have to systematically train it to, select one of the buttons that are lit up in a certain color and so on. Then the monkey finds out what the task set is, and you can actually really observe experimentally as the monkey discovers the different options, how the reaction times vary with that. And sometimes you can do that with humans too. Humans, of course, very often you can just explain what the task is, and you can actually also change the task once you can explain it, but then maybe suddenly targets of a different kind appear, and then people will adjust their computational process and thus their reaction time to these changes. And the third option is, again, with babies, where you don't explain, sort of like the monkeys in a way, except they don't have anywhere near as much patience. And so you can only do it a handful of times. And so just for the slide, I explained most of this. Important idea maybe to understand that the task set is by definition something that you know before the imperative signal comes. So we'll think of the task set as what we use the term pre-shaping, that is creating some kind of neural pattern of activation that is not necessarily homogeneous, because you already know something about where activation will have to be generated and have some representation for that. So the pre comes from before the moment when the decision, from which on the decision is made when you get the imperative signal. So in our terms, that would mean that the time course we're looking at is a time course from the arrival of a particular input that we'll have to model that specifies a choice, but it would be impinging not on a field in the resting state, but a field that already has some structure from the task set, which might be learned or might be by instruction, or you've just shown 5 buttons on the table and people just assume it's going to be one of those 5 buttons that's going to light up. And it's on that pre-shaped structure that then the input impinges and maybe ultimately creates a peak. And when the peak arises, you would respond, and where the peak arises would determine which response you give. In fact, the stimulus, we call this in the model specific input. It will be the model of the imperative signal. And it contains always 2 messages. It contains the message that you are supposed to answer now or make the response, and that you're supposed to know which one you're supposed to make. Actually, there's some paradigms that try to separate these 2 things, where you have a holding period, but let's gloss over that for now. So in the field, you can see that when you vary the task inputs here, assuming the task input could be 3 possible targets, as inputs, they would lead maybe to this green shape. This would be all still below 0, some threshold. And then at some point, you would perhaps apply a stimulus that selects the leftmost target, and as that happens, a peak might grow. And that would be enough input to push the system above detection. And so here is a more precise analysis of that to see the different factors in here. You would have the pre-shape, the expression before 0 here, before the imperative signal comes, and see a little bit of a local preparation here. And then you would have local stimulus. So different colors are different examples of giving the system the blue input, the red or the green. In the blue case, it would overlap with one of those three shapes, and the others are a little bit shifted from that. And you could also imagine that you're giving a homogeneous boost to the system, which would not specify any particular choice, but would be a pure go signal. So if you only had that boost, it would just say go. And so it might matter in the end of how much specific input you have, so localized input versus how much boost you have. A little boost, mostly specified, that would be a strong imperative signal. If you had only boost, that would be sort of like free choice, but with a go signal, where you tell the person when to make the response. See, when the local specific input is large, as it is here, then the field that arises from that clearly tracks the metric contents of the specific input. So here the colors match. You see that the field states here are sort of like the inputs, except there's a little bit of a distortion. You might see that they get a little bit attracted here to this location. So these two are a little bit closer than, for instance, the green and the cyan. The red and the green are closer than between C and R. A very subtle effect here, but actually can be observed. Such things can be observed. So with this kind of thinking, one can account for classical information processing kind of effects. It's kind of fun. We wrote a paper, and I'll give you an opportunity to read some of that about that. So this is a so-called Hicks law. The Hicks law says that the reaction time increases with a number of choices. Very intuitive, right? You might know that. So you have 2-choice task, then you're going to respond faster than if you have a 4-joint task or an 8-joint task. Typically, people double the number of choices because the curve turns out to be approximately logarithmic. So it's approximately straight line. It's actually a little bit saturates for large enough number of choices. So it's not a perfect logarithm. And the logarithm, as some of you might know, is the Shannonian measure of information. So, oh, wow, this is like information theory. There's also the Hicks law, where it's a question of the probability of the choices that determines reaction time. Number of choices, when the choices all have the same frequency of occurring, then 1 over the number is their probability. And you see that's a special case of the Hicks law. And Hicks law also predicts nicely the curve. And so people said, yeah, that must be the information processing number of bits. Now it does saturate, so that isn't predicted by that theory. And our model explains why it saturates. So in the model, it's a rather trivial consequence of this pre-shaping influence. Assuming you know the number of choices, you've learned that. That would mean that you have some way of pre-shaping your field. So in the model, we just made a schematic that there would be little peaks, like in 4-choice tasks, you would have 4 little areas that are a little bit pre-activated. It's all negative, sub-threshold, 8, 4, 2, or 1. There's actually a real difference between 1 and 2 that I'm glossing over here. But let's say from 2 to 8. In the plot, we're always looking at the case where the input was then supplied for the leftmost choice, just to make the reference the same. And then what is plotted here is the level of activation that is maximal in the field. It will be over this first choice after some time, as a function of time. And that goes from a negative value then ultimately to a positive value. So if you think that the motor ultimately would have to model the motor system that is really driven by that, but here we would just take some arbitrary threshold and say when that threshold is reached, that is when the response happened. Remember, I was arguing that you wanted cellular selection decisions. The fields actually will do that, and as a result of that, it goes very smoothly through the threshold, and there's no problem with measuring it that way. So, but that's just a shortcut, right? We will have to model the whole motor system if we wanted to do this better. And so what you see is that when the number of choices is larger, then the system rises later to the super threshold. So this is a question of parameter tuning. It has to be such that these inputs which are below threshold matter. They only matter if they contribute a little bit to interaction. So the sigmoid nonlinear function has to be a little bit soft enough to be sensitive to the fact that there are multiple locations. And then this really just comes from the fact that there is more overall inhibition in this case, because the peak building here gets a little bit of an inhibitory input from those other possibilities, and more so than a larger number of choices. And the subtle effect is that the effect saturates, because as you add more and more little peaks, they will also inhibit each other mutually, even though they're sub-threshold, a little bit. And so as you add more peaks, the net additive inhibition coming about is less, because the different peaks are already mutually weakened and therefore have less of an inhibitory influence on the peak which is going to grow. And we were able to actually really mathematically analyze that and sort of derive these curves from that kind of picture. But it's fascinating, but it's also boring, because this is like an old story, and you can also, in terms of information amount, seems like a post hoc story. So more interesting was perhaps something that would be really specific to the fields. The specific to fields is that we don't actually have these discrete choices here. They were just spaced so that the pre-shapes don't really overlap much, but that we might have real metric choices, perceptual or motor in nature. And when we have metric choices, then the metric distance between choices matters. And there's something paradoxical happening then, as you see here, when we have two choices, always two choice tasks, and the choices are far from each other, they, whatever is the pre-shaping influence will not overlap much between those two choices. And when the input signal comes, you know, either this peak grows or that peak grows, and you get a certain rate at which the peak level, you know, rises. Remember, you always have an imperative signal, so one of those peaks clearly grows and the other doesn't. But when they're close to each other, these inputs that make the pre-shape might overlap a little bit, and as a result, like this choice as part of an arrow is boosted a little bit by this neighboring narrow choice, so that it is at a higher level, and therefore if you now start to peak from there, you reach threshold earlier, and therefore there will be faster reaction time for the narrower choice, which is sort of counterintuitive, because you would think if the choices are more similar, just in terms of metrically discriminating them, it should be harder, and therefore it should take longer, and there's some empirical evidence for that too. So this is something that we thought was generic in the model, and we made some argument that reaction times in eye movements are like that. And I went to Marseille at the time into a lab run by Jean Requin, and he actually showed me that he had data like that. It was data that didn't make sense to them back then, because that was done a long time ago, way before I went to the lab. It was all data that were, you know, in the information processing paradigm. So this seemed a little bit odd, and they ended up publishing it in a French journal in French. So sure, a way to make it obscure, Le Travail Humain, you know, it's like the human factors journal, where had a very detailed, very nice study like that, and I was really ecstatic about that. I did cite that work, but it's you know, not very prominent. We were actually able to do some of our own experiments. This is with a student of my old collaborator, John Jaker. This is actually unpublished data, but actually a published work in a moment. It's an experiment in which we, very simple, you know, just so people have a stylus on a home button, and then they get some indication of where the target is, and they like two arrows, and they have to point at the end of this arrow tip on this whole, and you have to arrange that. When the stylus hits, you know, it measures the arrival time, and when the stylus lifts off the center button, that's when the reaction time starts. Well, you know, you measure the reaction time, and you have to do certain things to make sure that there's no attentional effect and so on. So that we were able to vary the metric distance between those two choices, either in a two-choice or in a three-choice task. That doesn't matter. And you see when the target separation is large, reaction times are larger, and they become shorter when the targets are closer. 90 degrees is a little odd here, but the shorter ones are clearly going down, or faster when the metrics are closer. Here's the more sophisticated version of that. For experimentalists like to do interaction effects in the statistical sense, because you don't really know the size of an effect like that a priori, and so the sensitivity to pick it up really depends on something substantially, over which distances will you see that. But if you have a known effect, and you can say that you can turn that effect on or off with another effect, then you have some kind of metric, because that known effect is sort of your instrument, how to measure that second effect. So that's what's called statistical interaction. So I'll make that concrete. So this is from the model. We would have two manipulations. One would be to vary the metrics of the two choices. The two choices here, they are far, here they are narrow. And the other is to vary the probability of the choices. So here in the model, reflected by how much more activation this pre-shape has than this here. This distance would be the difference in probability. And so you see more difference in probability and less difference in probability. In experiments, you vary the probability by having one of those targets appear on many more trials than the other one. So just statistically speaking, the frequency of the target across trials is higher for one than for the other. And that difference in probability you can make large or less large. And what you're exploiting is the Hyman law, that is the fact that reaction times are faster when the choice is more frequent. A rare choice, you will have a long reaction time. So in the model, this arises from this difference in level of activation. So if you select this more activated target, you will maximum activation will rise earlier because you're starting from a higher level than if it's less likely with this choice here. See, this is a small difference. If you take some threshold level, a small difference in reaction time. When the difference in activation is larger, it predicts a larger difference in reaction time. That's the account of the model for the Hyman effect, the probability effect. Now the interaction shows up here in that when the choices have the same difference in probability, but they are just pushed closer to each other. So in the pre-shape, they are no longer actually visible as bimodal distribution. So it's like this peak fuses into this peak, and that's why this peak here is larger, because it's the sum of these two. Then you're predicting that the probability difference now will become smaller again. So the interaction effect is, the Hyman effect is gone. It's gone by this low condition, the low probability condition, becoming faster. It's actually moved to the left here if you look more closely. That's the prediction. You see, we can work with this only because this is just pre-shape. There's still an imperative signal that will still make a peak over this choice or over that choice, depending on which one you're asking the participant to do. So here's the experimental paradigm. It's similar setup with the center target and then going out to some other targets. And so you had a wide condition where the one target was magically far from the other, and a narrow condition where they were close, and always one of those targets was showed up only about 1 every 10th trial, and the other was frequent, 9 out of 10, and it could be magically close or not. And the true experiment had symmetry conditions, like the frequent was also on the other side, and they were all randomized. And you also have to do special stuff to make sure that it's not just an intentional effect, that you don't get the wide target so well because it's so far from the focus of attention. So in fact, these gray bars here come from a condition where we check for that. So there's no attentional effect here. And here are the results for reaction time. So the red is the wide condition, and you see that the rare target has a larger reaction time than the frequent target. So that difference is the Hyman effect. And then the green condition are the same frequencies, same probability differences, but now they are magically close, and you see that the reaction times are equal, and they are actually equal at the level of the faster target, so the frequent target, except it's predicted by the theory. One can actually also do EEG with that and sort of map that onto that. That's all cool. So there's direct evidence for this magical effect. And let me check the time here. And that's very satisfactory for the theory. I think I want to skip this here and just very briefly do this one here. So the slides are on the webpage. I'm skipping those. Those are not so important. So just a brief comment on how to reconcile this with the intuition that magically close choices should be harder. And that's actually a known effect. It's sometimes called the distance effect. We call the other one a metric effect, just to make it a different word. So the distance effect goes back to the 1930s even. For instance, you're asking someone to pick the shorter of two sticks. And if the two sticks are very different in size, it's very trivial to do this very quickly. If they're very close in size, it takes longer to select the correct stick. So what's intuitively different about that is that it's clearly a categorical task per se. You're not supposed to reach like in the middle of the two sticks, right? You have to say which one is the shorter one. It's categorically, is sort of conceptually categorical. In that sense, it is sort of driven by pre-shape, right? The pre-shape makes the categories. And so a way to see that is if you had a pre-shape, which I made larger here, just for illustration, and you have a stimulus that now specifies which choice, but this stimulus is small in amount, and you're making the response by just giving a large homogeneous boost. So give a go signal, typical, give a categorization response, and give them just a little hint at which of the two choices is the right choice. Then, as you see here, they respond categorically. That is, if I put this local input with this large boost on top of this pre-shape, the fields that ultimately merge, but you see the different colors lie almost on top of each other. It's because this metric information is very small compared to the pre-shape. And so they make categorical responses. And if you now do the same manipulation about metric distance in this kind of situation, then you find the opposite effect. There is, I don't want to take you through this all, but if you go through this, and you can do this on the slides, you will find that here the opposite happens. That is, the metrically close choices have a larger effect of probability than the metrically far choices. And that's how one can understand how two different signatures can arise from the same theory. It's kind of interesting, right? This we never published, actually. It was probably in 6 months, and I didn't want to do that. I have to do that in some time. I rushed because all of this is sort of old stuff in a way, you know, just very classical stuff. In the paper, there are actually other things like stimulus response compatibility and the whole literature on that. The Simon effect is a whole literature. We covered that in just one paragraph there. Now, I'm more interested in this kind of thinking because this actually gets closer to embodiment. Now, you could say the reaction time is sort of like penalty shooting in soccer, right? You have the what do you call this guy, guard, or know what the word in English is, the goalkeeper, I guess, the goalkeeper standing there, and then the ball is shot and has to jump to the left or to the right, right? Guessing the corner. And that's of course not what soccer is about, right? That's just one very special case where you could say it's I'm standing there, I get information, and then I make a decision one time. There's a goal signal. When this guy gets close, then you have to jump. And apparently they do jump before they can even see the ball moving. So jump a little ahead of time because these balls are so fast. So I'm not a soccer expert, but that's what I hear. Now, of course, real soccer playing is much more dynamic, right? All the time you're seeing the ball, and as you're making decisions, you still see the ball, and you're as a response, as a consequence of your decisions, the information about the ball also changes because you're seeing the ball maybe from a different side depending on where you're coming from. So it seems like a very extreme limit case where you just reduce it to this one-time shot. And so a little bit more dynamic is this paradigm that goes back to the 1960s, was invented by someone called Brewer, or so Brewer. I don't know exactly how to pronounce him. I think it was maybe a Dutch fellow. And we became aware of it through the extensive work of Claude Guéz's group, who did this for somewhat different reasons, really. And we reinterpret this work now. This is already a long time ago. And it uses what we call the time movement initiation paradigm. It's not a well-known concept, but this has different names depending on who does it. It's a paradigm in which you can, in a sense, train participants to make a decision at a defined time. So what you do is you give a metronome, and then they learn to move on the fourth tone, for instance, and which of movement to make, you tell them by some stimulus, only a time interval before that fourth tone that you then vary. This is called a stimulus response time interval that you then vary exponentially. So when this is a very short interval that's shorter than a reaction time, than a typical reaction time for that chance, then they are guessing the corner, just like the soccer player, right? And when you make it large, then it's sort of like a regular reaction time paradigm. And that's the paradigm. I think here's a more clear illustration. A lot of this work uses isometric force impulses, like pressing on a stiff joystick to control a cursor on a screen, so to minimize the movement component, you can imagine. So for instance, in this experiment, there is, in this sense, target is a force level they have to generate on that joystick. There are 6 force levels that they are familiar with, they have learned. These dashed lines, like 3 up and 3 down, and they have to initiate a shift, a change in force level at the 4th tone of the metronome. So you see the pitch of the metronome goes up, beep, 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 and the 4th you do. And you have to train that. You see here that they do this. This is one single trace. You see, it's actually, this is an ideal case, maybe that's why it was shown, where really right after the metronome comes on, there is a excursion here to the correct targets. These are ballistic movements, so you just have to touch that level of force. And so when you learn which force level to produce, only this interval here before the fourth tone, and that is what is varied in experiment. So here, example data, this is where the movement went in the correct direction. This where it went in the wrong direction. Experiment, it was randomized up down, and this is when the top level of force, the middle level of the force, or the bottom level of force was requested. And to the right, you see this stimulus response interval that was imposed by the experiment. It's not, it's really measured from the stimulus to the response, and the response was not always perfectly on the metronome. It was a little bit of an effect. And so this is the true stimulus response interval. That varies from very fast, very faster than they imposed, like almost 0 to 350, 400 maybe. And the typical two-charge reaction time would be maybe 300 milliseconds, 250 to 350. So you see some remarkable things. First of all, you see directional errors for fast responses and not for late responses across the board. And then you see that when you're asking for a very large or very small amplitude, you see some transient from an initial default distribution, which is sort of in the center, to then the correct response. You see that in intervening times, they're actually making responses here that are never part of the request, right? They're making averaging kind of responses. And the same here, you see that there's some, here the cloud is clearly centered on the average between this middle force level and the final force level. If you're at the middle force level, you just see a little bit of sharpening. I see a little bit more crowded points here, but there's no systematic drift here. And you see the metric effect in here in these choices, even though then you never see these categorical mistakes for large choices. They had actually a whole discourse of how that is discrete versus continuous, how that matters. But this emerges just naturally from the model. And in terms of the model, what you're seeing is just the peak. It's like a stochastic measurement on the peak. It's on the subthreshold peak. So we produce this from the model. This is actually experimental results as histograms, and this is our model. And we inserted 0, which is impossible for the humans. But the short, medium versus long stimulus response interval captures as well. If you're giving the default answer, the average, then it's just a sharpening. If you're giving a one of the extreme answers, then you see initially the default answer in the center, and then it drifts to the sharpens and drifts to the correct response. And you see that matches the data pretty well. It's not complex data fitting, so it's a very limited number of parameters that play a role. It's very generic kind of results. We looked at a couple other versions. Here's a particularly nice one. This is one of the few cases where it was actual movement that was looked at, not just isometric force pulses. So people had a paradigm similar to what I just showed you before, our center out pointing task. Here they had 2 amplitudes and 2 directions, so 4-choice task. And the 2 directions would be narrow versus wide, and then in separate trials they actually also looked at different amplitudes and the varied metrics of the amplitudes. And so what you see here is that these are histograms of the responses. You see for the narrow separation, the early responses, like smaller than 80 milliseconds, are monomodal. It's simple. This is this narrow dotted line, is single monomodal distribution, and then intermediate it's a little bit broader, has some kind of inner structure, and then the solid line is larger than 40 milliseconds. You see there is a bimodal peak, so that means, and if you look at that, it is really correct responses. That is, they select correctly the targets. You see, metrically it's not quite perfect. This is where the choices are, and this peak is sort of attracted to the middle. If you separate the targets more, then the late response are very clear bimodal, and you see that the earliest already has a little bit bimodal, but this is sort of an averaging peak right in the middle. So for very fast choices, we have averaging, and then the peaks emerge. Now it's going to really go from averaging to bimodal So that's exactly what I showed you about the time course of saccadic decision making now observed in hand movement, and the time course is observed here by manipulating the time course with this intuitive signal. It's interesting because in the neural grounding lecture, which I sometimes gave at this point in time, and now I'll mention that later, you can see that actually you can see the population distribution in the monkey brain for movement direction is a broad distribution. And that is sort of in the same order of magnitude when it would start to go from monomodal to bimodal. So, and this is not very constraining because on thousands of different ways of how angular distributions can be structured, but it makes you think that you're really observing, in a sense, the population activation of these neurons in the behavior. It's an interesting perspective. Yeah, let's skip that. Good. I think that's just about right. In fact, this is my last slide. So I'm concluding that these selection decisions enable us to have behavioral signatures of the underlying decisions. And always of particular aspects of the model, metric effects, or these time courses of decisions. And I skipped over some, like probably you can always play off different things, probability against metrics and so on. And it ultimately, here the field, I mean, the decision-making field is just a vehicle. I mean, here we're not seriously probing that, for instance, decision is stabilized. That would only show up in the model if we really link it to a motor system and would look at, for instance, online updating that you keep making the same response when or tracking a target when the target changes while you go there, and there's some interesting stuff to be done there. So it's still very crude, but I would say the phenomenology of the sensory motor kind of cognition very nicely matches the concepts. And maybe this reflects it, the metrics of the tasks matter. And that's sort of interesting that the information processing framework has led people to think much more about these sort of higher level questions. For instance, there's a large literature on task switching, where it matters when you change the instruction what to pay attention to when making a response. But these are very subtle effects. They are reliable. But what you find is that when you change something about the feature values themselves, you have huge effects. A task can be hard or less hard in terms of the shapes and how similar the shapes are, or how difficult the colors are discriminated, and so on. These are large effects. And classically, people haven't been interested in those because those seem to be just an implementation detail of how it's done in the brain, how the nerves are tuned. But you see, in some sense, that is actually the neural hardware that makes these effects, and understanding them really means something. And I want maybe hark back to this difference between direction and amplitude. There was earlier work that's reviewed in this Elhagen paper you don't have a chance to read, in which someone called David Rosenbaum studied how the reaction time for movements varies when you give people different prior information. For instance, people have, let's say, fortress tasks, moving in one direction of a certain amplitude or another direction for different amplitude, just like in this paradigm here. In some cases, it was up and down, so it was 180 degrees direction difference. And then the theme was that it helps to know the direction much more than to know the amplitude. And there was a whole theoretical story about that, some kind of information processing thing that you first do direction and then amplitude. So if you know amplitude, you can't do anything with it because you have to first compute direction. So it was used to infer something about the computational structure of that preparation. Because there's a lot of evidence that amplitude and direction behave a little bit differently in movement, also in movement adaptation, as shown by Claude group, actually. That was something that people really resonated with. But it turns out in our kind of thinking, this doesn't have anything to do with that necessarily. This actually entirely emerges from the metrics of the choices, because what happens is that in direction, these choices were metrically very far from each other. They were pretty much in opposing directions, like 180 degrees. And then knowing that it's going to be the left or the right makes a huge difference in how far the pre-shaped solution is from the final solution. So no opposition from a opposing peak versus two peaks that are optimally competing. While in amplitude, the differences in amplitude they used were not very large compared to the sort of the width of those distributions. So they were in the averaging regime. So when you, in the averaging regime, if you know one of those two amplitudes, it shifts your peak a little bit, but because you're averaging it already, it's very similar in shape to the peak you will have when you then know the choice, and therefore it has a very small effect. So we predicted that when you make a large amplitude difference, it would also now start being advantageous when you make the amplitude. So you can see that asking these questions about the neural substrate, the underlying neural representations, the metrics and so on, can really give a different quality to the analysis. And that's what we would call a neural process kind of analysis. Okay, that's my lecture, and I'm going to switch to taking questions.